Hi, my name's Henry and I'll be your host for the latest episode of Inside Imperial, where we take you behind the scenes of the various facilities around the college. Today we are in the Sir Alexander Fleming building, where we're going to be looking around the facility for imaging by light microscopy. Thanks for tuning in, and please remember to add your questions below the stream. Okay, so without further ado, I'm going to go off now and meet the two staff members who very kindly volunteered their time to help us with today's broadcast. So let's just go over to the other side of the room quickly. Hello there. Hi sir. Hi. Would you like to introduce yourself to our audience?
Yes, please. Good afternoon, everyone. Hi, Henry and Martin. My name is David Gaborio. I'm a biologist and a staff member of the film facility at Imperial College, where we provide microscopes and training and assistance for all the researchers around the college for their microscopy experiments. So they come and chat to us about the experiments and the research project that they are doing. We advise them on developing the experiment, preparing the sample, and then we train them on the microscopes, and so that they get the best images possible for their research.
Excellent. And roughly how many different microscopes do you have here available?
So in the facility we have 11 microscopes. Mostly of three types. We have the wide field microscopes, where that one is an example of. We have confocal microscopes, which gives you that extra little bit of resolution. And next door later you will see we have a super resolution microscope to see things smaller than the limit of diffraction of light, which is amazing.
Wow, sounds exciting. I'm looking forward to it. So also you've got something on the go here behind you, right? Would you like to tell us a bit more about what we can see?
Yeah, of course. So this is let me start that live. Oh yeah, sorry. So here we are looking at cells which have been stained for mitochondria. So let me adjust the focus there. And those little wiggly things are the mitochondria, which are the little power units that live inside our cells. So those cells have been fixed, so nothing's moving, but if you had them live, all of them would be wiggling at a very high speed. So I can zoom in a little bit. You can see those little things there, which you inherit from your mother only. Only the mother pass on their mitochondria. And the cells have also been stained for another protein of the cytoskeleton called actin in another color, which I will show you in a minute. And the DNA, which you can guess sits in there inside the nucleus, has been stained with another chemical which will appear in cyan. So now if I find a nice cell that I like, for example that one, I think it's pretty good, I can adjust the focus, right, and I can take a picture in the free channels, which should appear in a second. Okay, beautiful. So let me put that over here. So this is the type of pictures that we would get from just a quick microscopy experiment, where you've got the molecules of actin, which form those filaments and support the strength and the structure of the cell. The nucleus here in the middle, and those mitochondria there wiggling around the cell. So those are fixed, but if you have live cells, you can also use our microscopes to follow the cells over time, right? So you can say I want, for example, a picture every 15 minutes, and you can leave them for up to 2 days, and you can see how the cells move, how the cells divide, how the cells react to maybe a drug that you put on them, or different environments, on how you culture them, and that tells people. That gives people a lot of information on how the cells function, really, in the context of like the normal body or in a disease setting.
So there are a lot of practical uses for this equipment then, clearly.
Yes, yes. Being here at Imperial is fantastic for collaborating with different research groups and lots of different model systems. So for example, at the moment we have collaborations in the field of lung development, looking at how lungs develop during embryogenesis, looking at project on malaria, project on how viruses infect cells, how plants respond to infection by parasites, and how you can design new molecules, for example, to cross the blood-brain barrier. So molecules that would actually enter the brain. So all of those things you can do with a microscope. I can show you a couple of examples of
please,
movies, if you want.
Don't forget to add your questions as well as we watch.
So with those microscopes, you can take movies in 3 dimensions, X, Y, Z, and once you have the X, Y, Z data set, you can then render it in 3Ds with different softwares, and that's a little animation just to show in gray we have the nucleus with the DAPI. Another video I've got prepared for you is a collaboration I have with Professor Peter O'Hare from the Department of Virology and his PhD student Eiki Sekin. And here we are looking at that little yellow spot is actually a virus that has infected a cell, and those magenta little spots are viral proteins that the virus has produced. And looking, so you have the nucleus again in cyan there, and you see the virus has entered on the side and has started producing those viral proteins. So looking at this and the relationship between that yellow spot and all the other spots tells us how the virus infects the cell, and once we know that, then you can get a better understanding of the process of infection and the disease.
Silly question again. We lost sound.
Oh, sorry. It seems like we momentarily lost sound. So, David, would you mind repeating again about the cilia? We had a question if you were ever using this to look at the cilia.
Yes. I don't have an image ready for you, but we are. The cilia is a very important organelle inside the cell, and you can probe it with an antibody directed against proteins that are making the cilia, and this is immunofluorescence, this is what we are doing here. And then you can highlight, once you got the right antibody, you can highlight any organelle that you want inside the cell really, and then take the pictures. It can either be fixed or in a live setting as well. It's nice to see the cilia moving around like this. And I was saying before also, you can pretty much see anything up to a size limit of about 250 nanometers, and then after that you need to go to a super resolution microscope.
And just out of personal interest, something I was wondering is, I guess this equipment doesn't come cheap. What's the kind of cost for something like this?
So you're talking between 150 and 300,000 pounds for a system like this. Yeah, so this is a high quality equipment. So
top of the range.
Yeah, we're very lucky with those.
Excellent. Thank you so much, David. Okay, so I'm going to stand back up now. And we're just going to go around the corner here and explore the rest of the facility. So please follow me. I'm just going to introduce you to the second member of the team, who is Andreas. Andreas, would you like to say hello to today's audience?
Yeah, hello everyone. I'm Andreas Bruckbauer. I'm the facility manager here at the Facility for Imaging by Light Microscopy. And I'm responsible to provide a good service for all our users, so that that things are running well, and also for the recovery of costs for the facility and the financial side. But I also sit with users and do training and advice on imaging. So I'm very much involved in the microscopy side of it.
And how long have you been at Imperial, Andreas?
I've now been here for 2 years, but obviously I'm in the field since more than 15 years in microscopy. And
could you tell me a bit about the facility and who's able to use it here?
So basically, all scientists from Imperial College London, including students, are allowed to use the facility. We also have a few external users from companies, but mainly it's Imperial College users, and we have around 300 active users in the facility, and a high turnover that we get like new users all the time, which are introduced to our microscopes.
So you must be a busy man.
Yeah, naturally. I mean, is what keeps us running, and it's good.
Excellent. So, what exactly are we looking at on your big screen here, Andreas? It looks very impressive. Would you be able to explain a little bit about what's in front of us?
Yeah, I mean, this is a super resolution microscope. David has touched this slightly. So instead of going like to 200 nanometer, which would be, I mean, if you think about a human hair, that's like 20 microns in size. And if you go 100 times lower, you are at the resolution of a normal wide field microscope, which is the one next to here. But with this microscope, we can go 10 times higher resolution and go down like 1,000 times smaller than a human hair. And I can just start the microscope here because it's live. And if I start the experiment here, and then you see all the little dots here on the left side. They're actually all molecules, and they are blinking, and that's a special trick here of this microscope. I just need to adjust here the display, because each blink is recorded, and then on the right side there is an image appearing, which then should have much higher resolution than all the individual dots on the left side. And it takes quite a long time to acquire these images. But I have one which previously is recorded. So you see that there are more and more dots coming here, but you can't really make out any structure yet. But if you do this for 15 minutes or so, if I go here, so then you see here, this is actually tubulin on cells. So these are all little strands of microtubules giving structure to the cell, and that's basically a 2D image here of this with high resolution, and this can be down to 20 nanometer or even better. But it all works by blinking of individual fluorophores, individual molecules, dye molecules, which are laser illuminated, and then do this trick. The technique related to this has won a Nobel Prize in chemistry in 2014, so that's a quite recent development. And I mean, we are lucky to have one of these microscopes here at Imperial.
Yeah. Wow, that is impressive. Yeah, and the people who use the facility are very lucky to have such access, aren't they? So I noticed that also behind you there, of your shoulder, we've got a very large piece of equipment that I've been looking at ever since we arrived. This seems like the showstopper, so to speak. So could you just introduce us to what we're looking at here?
That's the actual microscope which records the images we just seen on the screen. So if I open like a few flaps here, you might see like in the middle there is actually the sample, so it's a slide, and you see there's a little circle is in the middle. There is where the cells actually are. And then we have the whole microscope, so it's still a normal microscope where you can look through the eyepieces and see things through here. But then we have a whole range of boxes on the right side where the lasers are in and the equipment to actually do the special effect and get it to blink.
And I believe it has a particular name.
This is our ELIRA super resolution microscope. So ELIRA is a double star system, and it kind of lends the name because of the technique. So that's what the company has named it.
I see. And again, just because this kind of thing interests me, what's the rough cost for something like this?
I mean, this comes close to £500,000, so that's a bit more expensive than the normal microscopes. Yeah, it's a lot of money.
Yeah, it's quite the investment. Excellent. Do remember to keep adding your questions below the stream if there's something you'd like to know while we've got both Andreas and David here. I'm just going to ask you a little bit more about how long the facility has been on campus as well, as you're the manager.
So roughly about 12 years. So some scientists came together and said, "Oh, we combine our resources, our microscope we already has, and then they applied for grants to get additional microscopes, and then it all started. And since then, it's running well."
And we're just in one small part here, aren't we, of the facility today? There is actually more rooms, that's right. No?
So this is one microscope room out of 5 rooms, and we are spread over 2 sites. So we have microscopes here in South Kensington and at the Hammersmith site. So it's 11 microscopes altogether, and hopefully more in the future.
Excellent. Thank you so much, Andreas. Okay, so there you have it. Thank you so much to Andreas and David for giving up their time to give us a tour today. If you're ever on campus, please do drop by and try and look around the Sir Alexander Fleming building. It is one of the most impressive sites on our campus. Don't forget to like our page and share this stream so that your friends can also get a glimpse of what goes on inside this facility. It's a bit of a mouthful, but I'll just repeat the name for you. It's the Facility for Imaging by Light Microscopy. So please check that out online. Thanks again for joining us all today. I've been Henry. We're going to say goodbye now and sign off until next time for the next episode of Inside Imperial.